So today we're looking at DNA, genes and chromosomes. So, how does DNA in prokaryotic organisms differ from the DNA in eukaryotic organisms? What is a chromosome? How are genes arranged on a DNA molecule? What are homologous chromosomes and what is an allele? So, in prokaryotes, the DNA molecules are much smaller and are circular, and they are not associated with proteins or histones. So they're smaller, circular, and not associated with proteins. In eukaryotes, the DNA is a lot longer and it's a linear molecule. It is packaged into chromosomes just before cell division by associating with proteins or histones. And then the molecule becomes supercoiled. So now looking at the mitochondria and chloroplasts DNA. The mitochondria and chloroplasts of eukaryotic cells contain DNA which, like the DNA of prokaryotes, is short, circular and not associated with proteins. So if you're ever asked a question about what the DNA is like in mitochondria or chloroplasts, just think of prokaryotes because it's similar, it's short, circular and not associated with proteins. Okay, now we'll look at the chromosome structure of eukaryote cells. The chromosomes are obviously found in the nucleus. Now chromosomes are only visible, they only become visible in prophase, so in the start of nuclear division. Most of the time, the chromosomes are just widely dispersed in the nucleus. When visible, though, the chromosomes appear as two threads called chromatids, which are attached by the centromere. DNA in chromosomes is held in position by histones. So the very long DNA, which is about 2 meters in every human cell, is highly folded and coiled. So it wraps around these histones, these proteins, and then it gets folded and coiled and looped, and then it coils in to form the chromosome structure. So you can see here you've got the double helix, then gets wrapped around histones, it gets coiled and looped and supercoiled to form the chromosomes. So DNA is a double helix which is wound around the proteins or histones to fix it into position. The protein complex, the DNA, is then coiled, and then it's coiled further and looped before being packed into chromosomes. So there's a lot of DNA which is condensed into a single chromosome, into a very small space. And each gene occupies a specific position or locus on a DNA molecule. So again, you can see here you've got the DNA molecule that then gets wrapped around histones, it gets coiled and looped, and then it gets coiled and packed into the chromosome structure. Now, in humans, human adult cells, we have 23 pairs of chromosomes. So the first 22 are known as autosomal chromosomes, and the 23rd is known as the sex chromosomes, which can either be XY in males or XX in females. So, now we'll look at homologous chromosomes. Now, the number of chromosomes and each member of the same species will be the same. So, for instance, all humans have 46 chromosomes. However, each species has a different number of chromosomes. So dogs, for instance, have 78 chromosomes. So you have to be careful, make sure you read the question. Don't just assume that it's 46 like in humans. Most species have an even number of chromosomes in adult cells because homologous chromosomes are pairs of chromosomes. So therefore they have an even number. Now, sexually produced organisms result from fusion of the gametes, so for humans it would be the fusion of the sperm and the egg, which then contributes a set of each of the chromosomes to the offspring. So you get one of each pair from the mother, the maternal chromosome, and one from the father, which is called the paternal chromosome. So homologous pairs are where you have one from the mother, one from the father, and that forms the diploid number in humans, which is 46. So diploid means they have the full set of chromosomes. Haploid is where the cells only have half the number of chromosomes. So sperm and egg would be haploid. They only have 23 chromosomes. But adult human cells have the full number, the diploid number, which is 46. A homologous pair is two chromosomes that determine the same characteristic but may contain different versions or alleles. So you can see on this chromosome here, they might have the same genes, so that's the gene for blood group on the same position or locus of the chromosome. You might have the gene for eye colour here in the same position, but there might be different versions of the alleles. So this might have an allele for blood group A, an allele for blood group B, but they're the same gene, they're both the blood group gene, but they have different versions. Or the gene for eye colour might be here, but there might be different versions. You might have blue eye allele on this one and brown eye allele on this one. In meiosis, halving chromosome number ensures each daughter receives one chromosome from each homologous pair. The cell receives one set of information for each characteristic, and when the haploid cells combine to form a diploid with the paired number of chromosomes, the chromosome number is restored. Alleles are different forms of the same gene. So homologous pairs of chromosomes have the same genes but have different alleles. So you can see here that this is the gene for flower colour, but it might have the allele for purple flowers and the allele for white flowers. So mutations are differences in the base sequence of a gene, and they may result in a non-functional protein such as an enzyme. So the DNA contains the genes for making proteins which determine the characteristic, and the sequence of bases determines what amino acids are coded for, and if the bases change, this base sequence change, it might code for a different sequence of amino acids, which then means that the protein will fold differently, the tertiary structure will be different, which means the protein may be non-functional. So in summary, in prokaryotic cells, DNA molecules are short, circular, and not associated with proteins. In the nucleus of eukaryotic cells, DNA molecules are very long, linear, and associated with proteins called histones. Together, a DNA molecule and its associated proteins form a chromosome. The mitochondria and chloroplasts of eukaryotic cells also contain DNA, which, like the DNA of prokaryotes, is short, circular, and not associated with proteins. A gene is a base sequence of DNA that codes for the amino acid sequence of a polypeptide, a functional RNA, including ribosomal RNA and tRNAs, and a gene occupies a fixed position called a locus on a particular DNA molecule.